Hello everyone, this project is about developing a drug which is going to have potential to cure cancers like pancreatic cancer, lung cancer, melanoma, acute myeloid leukemia, thyroid cancer, and colorectal cancer. These cancers occur due to mutation in a signal transducer protein called as RAS. RAS stands for rat sarcoma. RAS acts as one of the signaling proteins which transmits the cell growth and cell proliferation signal downstream. RAS is a G protein. That means it is linked with guanosine diphosphate. So when it is linked with guanosine diphosphate, RAS remains in inactive state. So when guanosine diphosphate bound RAS is replaced with guanosine triphosphate, it becomes active. And in this process, there is a nucleotide exchange factor that is called as SOS protein that is involved in this process. SOS replaces the guanosine diphosphate bound RAS with guanosine triphosphate. This SOS protein is recruited by another signal transducer protein that is called as GRB2, growth factor receptor bound protein 2. So guanosine triphosphate bound RAS is the active state of RAS, and it transmits the cell growth and cell proliferation signaling downstream by activating MEK and MAPK signal transducer proteins. RAS has intrinsic GTPase activity. That means it can dephosphorylate and hydrolyze the guanosine triphosphate, the bound guanosine triphosphate, into guanosine diphosphate. After transmitting the cell growth and cell proliferation signal downstream, what guanosine triphosphate bound RAS does is that it inactivates itself by hydrolyzing the bound guanosine triphosphate and dephosphorylating the bound guanosine triphosphate into guanosine diphosphate by the help of a protein that is called a GAP, GTPase activating protein. Upon the hydrolysis of guanosine triphosphate, the guanosine diphosphate RAS becomes inactive and the cell proliferation and cell growth signal terminates. This RAS protein is synthesized by RAS gene, which is a proto-oncogene. So when the mutation in the proto-oncogene RAS gene takes place, the RAS oncogene synthesizes the mutated version of RAS protein. The mutated version of RAS signal transducer protein lacks the intrinsic GTPase activity and it becomes insensitive to GAP, GTPase activating protein, the protein which was assisting in the hydrolysis of bound guanosine triphosphate with RAS into guanosine diphosphate, inactivating RAS protein. But since the mutated version of RAS is insensitive to GAP, therefore GAP cannot hydrolyze the bound guanosine triphosphate and the RAS protein remains in active state. So the mutated RAS cannot inactivate itself after transmitting the cell growth and cell proliferation signal downstream, and it continues to transmit the cell growth and cell proliferation signal. So it causes uncontrolled cell growth and cell proliferation, and it ultimately results into the cancer known as the RAS-driven cancers, which I have discussed earlier. The drug which is going to be developed under this project will target the guanosine triphosphate and the guanosine triphosphate bound domain of mutated RAS protein, defunctionalizing and distorting the guanosine triphosphate and the entire mutated RAS protein, terminating the uncontrolled downstream cell growth, cell proliferation signal. The drug development is based on FBDD, fragment-based drug design technique. We have selected some fragments which have very high affinity for the mutated RAS proteins, especially the domain connected with the guanosine triphosphate in mutated RAS. After that, we have selected a reactive group which targets the phosphate group of guanosine triphosphate, distorting it, defunctionalizing the guanosine triphosphate. The interaction of the selected fragments with the guanosine triphosphate bound domain of mutated RAS protein and the nucleophilic attack of the selected reactive fragment on the phosphate group of guanosine triphosphate causes mutated RAS inhibition. We have also introduced a protective group in the drug molecule, which will prevent the drug molecule to interact with the non-target proteins and molecules. All the selected fragments are attached and assembled strategically to design the final drug molecule. The drug molecule is designed to target mutated version of RAS protein. So I'm going to tell their PDB IDs, protein data bank IDs. So they are 7ROM, 7RON, 7ROQ, 8BE3, 7VVZ, 8VM2, 6ZIZ, and 7T47. We have conducted some computational procedures like molecular docking and molecular dynamic simulation to check the affinity of the drug molecule with the mutated RAS proteins, and also to check the stability of our drug molecule. So the result is that the drug molecule shows a very good affinity towards the mutated RAS proteins, and the molecule is very stable. Overall, in this limited computational test, the molecule is very promising. But we need to conduct more computational tests, and that's going to be possible by your support, because we are going to conduct the computational tests on very large scale to test the drug molecule. And in this process, we may replace the selected fragments with some other fragments which may have greater affinity towards the mutated RAS proteins. After we will have the successful computational tests, we will train a machine learning model which will predict the possible retrosynthetic route for the laboratory synthesis of this drug molecule. And for this, we are going to hire a team of AI developers. After we will have the possible retrosynthetic route predicted by the trained machine learning model, I will hire the team of organic chemists who will develop this drug molecule in the laboratory. And that predicted retrosynthetic route by the trained machine learning model will be very helpful for the organic chemists to develop the drug molecule in the laboratory. And when the drug molecule will be finally designed, we will get it tested by the help of organic chemists and the biochemists on the mutated RAS proteins in the laboratory, and also on the genetically engineered rats made to suffer from RAS-driven cancers. So this project needs money and time, So by your support, I wish this project is going to come true and this molecule is going to come true. And you can help, you can donate to bring this new discovery, new invention in the field of cancer research. Thank you so much.